So welcome to today, module 7 in our series, and this one's going to be about the human gut, the gastrointestinal system, or more specifically, we'll call it today the human biome. So just to remind us of the surface anatomy first of all, so looking from the front of the body, the sections here in the middle, in the upper part, is called the epigastric region, just below the xiphoid here. And then the right hypochondrium over the liver area, and the left hypochondrium, more the sort of stomach spleen area. And then in the middle section here, we've got the umbilical area, obviously around the umbilicus. And to the right and the left, we've got the left lumbar and right lumbar areas. And the lower part here above the pubic symphysis is the hypogastric area, with the right and the left inguinals. So those are the different quadrants on the anterior part that we have a look at as far as the abdomen is concerned. Now, a lot of people have gut pains, abdominal pains of various forms, and what I've done here is I put into each of those quadrants the sort of conditions that we get. So the right hypochondrium would be gallstones, stomach ulcers, pancreatitis. The epigastric is the stomach again, heartburn, which we're going to be talking about, indigestion, again pancreatitis, gallstones, and epigastric hernias or hiatus hernias. On the left-hand side, we've got the stomach ulcers, duodenal ulcers, biliary colic, and pancreatitis. So you see there's a certain amount of moving over of conditions, isn't there? And we need to be quite accurate with our diagnosis. If a person has pain, it can be being referred from, sometimes another quadrant there. On the right lumbar area, we've got kidney stones, urine infections, constipation, and lumbar hernias. And on the right side, we got, again, similar diverticular disease, constipation, inflammatory bowel disorders. And around the umbilicus, again, pancreatitis, early appendicitis, stomach ulcers, inflammatory bowel conditions, and umbilical hernias. And then we've got down in the inguinals, the appendicitis, constipation, pelvic pain, that would be more sort of gynecological, and inguinal hernias. Similarly on the left side there, and in the middle, on the hypogastric area, we've got urinary infections, appendicitis, diverticular disease. So one of the first questions I always ask people when they come in and they've got abdominal problems is, when do you get them? And are they made worse by certain activities? And so if a person has abdominal pains when they wake up, and this is particularly common with children because children often complain about tummy aches, and you never know whether it's because they want to get out of school because they've got an examination, the test that day. So on waking, this is important, that if you get problems, abdominal pains on waking, think parasites. If it's immediately on eating, then they're eating something which is having a reaction, like a histamine reaction in the stomach straight away. So if they eat something and then they get tummy ache. If it's something is relieved by eating, it's usually because it absorbs the stomach acid, and therefore it's probably a gastric ulcer, is the likelihood there. If it's 10-20 minutes after eating, in other words when the food's in the stomach and it's then not being digested, it could be low hydrochloric acid. As we'll see, hydrochloric acid basically goes on to activate pepsin, the proteolytic enzyme in the stomach. So it could be low hydrochloric acid or low pepsin. And if it's after that, 1 to 2 hours after eating, and the food's down into the small intestine by that time and it's not being digested, it could be pancreatic. So in other words, a lack of digestive enzymes there. So that gives you a rough idea, if you ask the patient, when it is that they get their tummy aches. Now, from our point of view, we're going to have a look at the different areas of where we might therapy localize from a kinesiology perspective. And there's a couple of areas which are used a lot in applied kinesiology. Is the ileocecal valve is one of these, which, as we'll see, is halfway between the anterior iliac spine here and the umbilicus. And the other one is the diaphragm for hiatus hernias. So these two functional problems are two of the most common things that we come across in clinic. The pyloric sphincter, particularly important with pyloric stenosis in newborn children here. The sphincter of Oddi, which is where basically the common bile duct with the pancreas comes in to exude the liquids into the small intestine. The flexure of the large intestine around the liver, the hepatic flexure, and the splenic on the opposite side. The junction between the duodenum and the jejunum in the small intestine here, it's like a little kink shape there where wind and things can build up like the flexures here. The ileocecal valve, and on the other side, controversial, is what's called the valve of Houston, which really is the junction of perhaps the descending colon to the sigmoid. There seems to be a slight flexure there. Other people call it down here between the sigmoid and the rectum. It's a little controversial whether it exists at all. Gray's Anatomy does actually say that this is the area of what's called the valve of Houston. Whether there is a valve there or not, we're not sure. It may be just a switching or neurological disorganization of the other side. So those are the main points. And this other one, which is halfway between the umbilicus and conception vessel 6, is here. And then you've got the TSB of the triple warmer, small intestine, and bladder meridian points here. So just about 2 inches, 5 centimeters inferior to the umbilicus. We use this therapy localization point with pressure to see in the root of the mesentery, and this seems to check for the need for probiotics, which we're going to talk about later on. But it is with pressure. It's not just therapy localizing to it, but if you press into the root of the mesentery, it's often a need for, seems to indicate a need for good bacteria. So these 2 points are very common. These are the flexures, as I say, of the large intestine. So on the hepatic flexure, we've got the ascending, transverse, and then the splenic. And because we've got 90-degree bends in here, and sometimes more than that with the transverse sagging down, this is where wind gets caught. So you get a lot of build-up of flatulence in those areas. And there's the conception vessel 6, that point for the root of the mesentery. So we do a kit with all the sort of test substances that you're going to need, the biomarkers, and we divide it up now into the newer kits in color coding. So we've got the digestive enzymes here in blue. Then we've got pathogenic bacteria, including the parasites like nematodes, cestodes, etc. on there. And then you've got the volatile organic chemicals that can be produced in the gut, which we'll talk about, particularly things like ammonia, phenol, methanol, aldehydes, etc., and the short-chain fatty acids. And then we've got naturally occurring food toxins. These are the things that are naturally occurring substances which can be toxic, not allergies, but substances which can be naturally containing, like isothiocyanates, etc., within cruciferous vegetables. And then we've got probiotics, extensive list of 22 different probiotics here, and prebiotics, which we'll talk about, are the food that the probiotics eat and proliferate as a result. So that's the gut kit. Now, what we're going to do now is a scale. So if you cut there back, Gary. Okay, because scaling, doing verbal challenges is not an official ICAK, and a lot of other schools don't like it either. I find it's the only way of really actually testing something that you can't really evaluate in any other way apart from talking to a person's subconscious. Yeah. So the biome actually begins in the mouth, as does all digestion. So digestion, as we'll see with alpha amylase as the enzyme in the saliva, but start in the mouth, and many people count the mouth, and particularly the tongue, as the dipstick of the intestine. So what you see in the mouth is a reflection generally of the intestine, and so it's a reflection of the state of the intestine is the state of the mucous membranes and things in the mouth. So we know that teeth are an important giveaway of general health. Here we see healthy gums, and here we see gingivitis, or inflammation of the gums. So this is the red area around here. and then eventually the teeth become wobbly and loose and maybe develop plaque around here, but you can see the opening of a pocket here where food and bacteria, of course, can get down into there. And remember that more people now lose teeth from periodontal disease than they do from dental caries. And then you see advanced periodontitis with the exposed root, and eventually you get loss of bone. So when you have a look at somebody's teeth, you want to have a look around there, and you're looking for the line of the gum as well as the colour of the gum, if in case it's red and things. But if you can see a receding gum, which is very common here, that is the first sign, if you like, potentially of osteoporosis. So if you see a receding gum, think this is not just a receding gum, and they may need some special nutrition. But what I would do is also think that they could be losing bone mass. May not be now, but a little bit further down. So generally, cavities depend upon the state of the enamel. So if your enamel is good, bacteria and things can't get through there. But obviously if the enamel is wearing out or it's cracked. And the enamel is made of many different minerals, but the prime one is calcium fluoride. It's a very important part of the fluorooxidate which makes up the enamel. But it's not sodium or tin fluoride. The natural form of fluoride, which is present in a lot of plants and things and water, is calcium fluoride. There's an area in Texas in America, the Smith something county, I've forgotten the other word for it now, which is a whole district there where they have no dentists because nobody gets dental caries. And they studied it for years and years as to why it was. And it was that area that they decided or discovered that it was a naturally high fluoride area, and it was that they based then fluoride as the solution to reducing dental caries in everybody and putting fluoride into water where the water level, natural water level of fluoride was low. So there's some areas in the country where fluoride is at a normal level, but it actually has made no difference to dental caries. The incidence of dental caries is the same whether it's fluoridated or not, which they've had a long time before they've actually admitted this. But most of the natural nutrition, like Weston Price, etc., who study this intensely, say it's not fluoride, it's manganese. So manganese is the key element within the bone mass and also the teeth. For the fluoroacetate composition is actually manganese is important. And this area of Texas, the Smith County there, is naturally has a high level of manganese. So think manganese, again with dental caries. Wobbly teeth is a sign of collagen problems. And collagen, as we're going to learn, depends a lot upon vitamin C to hydroxylate proline and lysine. So this is how Linus Paulding won his first Nobel Prize on the structure of collagen and the importance role of vitamin C in the integrity of collagen. And again, with collagen, we need manganese. So manganese is an important cofactor in that synthesis of collagen. So wobbly teeth, think vitamin C, manganese. Receding gums, the gums are the area which replace the cells every 20 minutes. So every time you lose a cell, you grow new ones every 20 minutes, okay? So every time you have a piece of toast, you probably lose millions and millions and millions of cells in your mouth. But by lunchtime, you know, your gums haven't receded. So it's the first place that you see folic acid deficiency, or folates rather than folic acid deficiency, is in the mouth. So if you see those receding gums, think folates. Now for us, it's nice and easy because you can therapy localize the receding gum because it'll therapy localize, and then you can test the nutrients that you see on your chart there. But remember, it's not necessarily folic acid as such, because that's an inert vitamin. It doesn't have a function in the body. What has the function is the activated forms, the methylene, methanol, and 5-methyl-tetrahydrofolate. So you will need to test with the activated forms of folate and not just folic acid itself. CoQ10 is recommended by a lot of dentists. This is to do with more the energy of the cellular structure. And then you notice that they're putting CoQ10 minuscule amounts into some toothpaste because it's a good sale point of view. And the old biochemical tissue salt, silica, is very important. Silica is a substance in the ground matrix, which we'll see is collagen and cartilage in joints and things. And it's a crystalline sort of structure, and it builds the crystals of bone and of collagen around it. So silica is very important. Silica, the best form of silica used to be horsetail equisetum, which is that plant grown on seashores or sandy sand dunes. But bamboo is the better one, really. Bamboo, it's the pith inside the bamboo, which then becomes the hardened part of the actual stem, the outer part of the bamboo itself. So from the pith of the bamboo, it's something like 70 to 80% pure silica. Because silica otherwise is glass or sand, and it's insoluble. So it doesn't matter how much sand you swallow, you're not going to get any silica in. You've got to have it in a soluble form. Bleeding gums is usually a sign of high homocysteine. In other words, again, low collagen within the blood vessels. And to metabolize homocysteine, we need pyridoxal 5-phosphate to convert homocysteine down into cysteine. So that's pyridoxal 5-phosphate, the activated form of B6. So one of the first places you see B6 or pyridoxal 5-phosphate deficiency is in bleeding gums, what they call the pink toothbrush. So when you look at the toothbrush, or you spit it out and you then start spitting blood out, it's because the gums are beginning to bleed. And this is very common in pregnancy, the first few months of pregnancy. And this is why a dentist can often tell you, or tell a woman, you're pregnant, aren't you? And she'll say, oh, I don't know, because the gums are bleeding. Okay, it's one of the first signs of pregnancy. And you can guarantee that a girl who has bleeding gums during the pregnancy will have a lot of morning sickness. Okay, because morning sickness is due to a deficiency, as we talked before, of pyridoxal 5-phosphate. So look out for royal pregnancies and the relationship you'll see of morning sickness in certain royal families, or certain members of the royal family. Can be folates as well, because again, folates will allow tissue not to regenerate as quickly as that, so you get receding gums and sometimes bleeding. And also certain bioflavonoids, which are more involved with the permeability and strength integrity of the capillaries and things. And the most important one there is rutin, which you find a lot in buckwheat. So rutin, buckwheat, is a good supplement, or to use buckwheat within foods as an alternative to wheat. Plaque is a definite build-up of bad bacteria in the biofilm. So this is usually an indication that the person needs some form of oral probiotics. So we're going to look at probiotics in detail, but it's determining which are the ones that this particular person needs. So we need to tailor the type of probiotic or the forms of probiotic for the person. But plaque is definitely a bad bacteria. And of course, a lot of these people use mouthwashes with antiseptic in, so they're basically killing all the good bacteria as well as bad bacteria in the mouth. Bad breath, reflected bad, also usually is an indication of a gut problem. Okay, people always think it's a lung problem, but most often bad breath is gas being produced in the stomach and the intestine coming back up, and is usually an indication of a gut problem, which means probiotics again. Gingivitis, inflammation of the gums, we need to have anti-inflammatories. Vitamin C here, rutin. B complex is important. And I think I'd add in there clove. We've talked about clove before, but clove is very anti-inflammatory, as is turmeric, but clove is actually a lot more anti-inflammatory than turmeric. Tartar is a build-up of calcium, usually from the different salivary glands, that the saliva is high in calcium, too high. And so here, to shift calcium and to get the balance, we need the opposite, which is magnesium. So calcium's outside the cell, magnesium's inside the cell. So people need more magnesium. They need oils to get the calcium back into solution. And nowadays, vitamin K2, we know, takes calcium out of tissues where it's deposited and puts it back into the bones and the teeth where it belongs. So those three things will be very important with someone with tartar. Mouth ulcers, we've learnt through the years, the hard way, are pyridoxal 5-phosphate deficiency. So pyridoxal 5-phosphate produces mouth ulcers. Occasionally it's a sign of folate, again, that you're not regenerating the cells. You bite your tongue or you bite your cheek, and then you get this ulcer because it doesn't repair properly. It can be folate, but 9 times out of 10, mouth ulcers, like all ulcers, are pyridoxal 5-phosphate deficiency. And finally here, when you have amalgams of the metals removed, it's a good idea to have something which conjugates onto the metals. And one of the natural ways of doing this in the body is selenium. So selenium as a liquid, used as a mouthwash, not swallowed, but used as a mouthwash, will bond on to particularly mercury, and then you spit that out after the extraction with the dental hygienist. So selenium as a liquid. Charcoal is a good blotting paper, again, of toxins, as is bamboo. Again, this is the source of the silica that we talked about. Chlorella, spirulina, all things that act as blotting papers for toxic metals in the mouth. So this is a research that probiotics may improve the chances of avoiding dental caries. Well, that's probably fairly obvious by now. So amalgams, just to refresh ourselves, are made of different mercury, different metals, mercury being 50%. So it's a bit ironic that they call them silver amalgams when in fact the vast majority is mercury. Silver could be as high as quarter to nearly a third of it. 14% would be tin, less than 8% copper, 2% zinc, 4% indium, and 0.5% palladium. The reason for lower mercury emissions is that amalgams prepared with indium rapidly form indium oxide and tin oxide films, which reduce the mercury release. Palladium reduces the tarnish and the corrosion. So you can see that the reason why they make it an amalgam, in other words, we mix other metals in, because during the years they found they can actually reduce the emissions and leakage of mercury by putting other things in, like the indium and the tin. But they could be toxic in their own right. So the half-life generally of a silver amalgam is about 10 years. So if a silver amalgam lasts in the mouth more than 10 years, it's probably going to be pretty stable. But if it comes out, it's going to come out because if you're losing the mercury, your amalgam is shrinking, and then eventually it will just drop out. And that's a sign that if it drops out, it means you're leaking, and you've been leaking, and where's the mercury go? Possibly into you and other locations. So having had the amalgam removed, it's a good idea then to use something which flushes metals out, such as black walnut, coriander, or coriander herb or spice is good, lemon balm, alpha-lipoic acid, yarrow, N-acetylcysteine, because that goes on to make glutathione, which is the main metal detoxifier, taurine, zinc, silica, sulfur, shark liver oil, and again charcoal. These are all good chelators of metals in the system. So part of, if a person has a toxic metal problem, part of it is being able to remove the amalgams here, but it's then trying to detoxify the metal from inside the body. It's important to look at mouths because if a person has a dysbiosis, in other words, a problem with the bacteria in the gut, which sets up all these problems, it may be that the good bacteria are being killed by mercury or other metals being leaking from the mouth. So in other words, you could spend all your time putting good bacteria into a person, giving them all the digestive enzymes, but then you find that you're just poisoning it because they're actually leaking amalgams. And luckily we don't see it so much because more and more people are not having mercury or silver amalgams put in anymore. But there are still quite a lot of people who've got old ones in there. But very few people go to the dentist and get a silver amalgam put in now, luckily. So the biome. The biome means basically the microorganisms that live within us. So there are innumerable living organisms in the human gut which play a vital role in maintaining good health and well-being. And it may very well have an important impact even on the lifespan of an individual. Back in 1600, the oldest living person was 119 who died, and that when they dissected him, which they did we were allowed to do in those days in 1600, only just, they found the colon was as clean as a whistle. And that's the comments that the pathologists made at that time. There was no caking up of the colon in any way, that the intestine was completely clean in this person. So if we imagine that, if you've ever had a colonic irrigation, or people who have colonic irrigation, what comes out is quite disastrous with all the fungus and the parasites and everything else. So having a good clean gut is an important part of also having a good clean and long lifetime. The human microbiome is the full complement of all the species that live there. And of course, inside those bacteria, they have their genes within them. So their genes and the genomes that inhabit the human body. In a healthy human body, the microbial cells in our gut outnumber us 10 to 1. So there's more microbes in our gut than there are cells in the human body by at least 10 times. So why is our gut not so big then? And the answer is the microbes are very small. Okay, the bacteria are much smaller than human cells, which is why you can pack in so many more times. Present on all exposed areas on the surface of the body, so outside, inside all the mucous membranes, throughout the whole body. The greatest diversity and density of these different microbes is in the gut. The human biota, in other words, all these microbes contain greater than 100 times more genetic material than our own. So within the genetic material of the microbe are every possibility of every permutation of the genetic code times 100. So in other words, there's many different types of genetic makeups that we don't even have. So within those microbes is the answer to all our human problems within our genetic expression. So if we have a problem with a gene that's not functioning properly, perhaps we could find a microbe that's got the same one and pop them into the gut. Because they know with fecal transplants, people can change completely. If you put in the right bacteria into a person from somebody else's bacteria, their health changes dramatically, as we'll see. Microbiome disturbance is due to the food that we eat. The main one, which is probably much more important than anything else, is food preservers. Food preservers are in everything that's packaged. Everything to extend the shelf life as a preservation chemical will destroy the microbes in that food and destroy the microbes in your gut. So food preservers are much more important than things like antibiotics. The lack of fermented foods, we don't eat as many fermented foods these days, that we used to use fermentation as a way of preserving foods for the winter. We use a lot of emulsifiers in the foods. Antibiotics, not just medicinal, but within animal feed, particularly chickens and things. Over hygiene effects, detergents, mouthwashes, chlorinated water. The idea of chlorine is, of course, hypochlorite is to destroy microbes. And environmental chemicals and antiseptics and in toothpastes. So let's just have a look at the gut kit again with the chemicals. So optimizing the gut, we need to think about hydrochloric acid, particularly for those people who have digestive problems, who don't digest all their food. Hydrochloric acid and pepsin, which is the first enzyme of digesting proteins. The pancreatic digestive enzymes, which then are activated by small intestine enzymes. Bile salts, which help us emulsify fats. And then we've got our bad guys like the fungi, who can withstand all sorts of imbalances in the environment, but it's usually the imbalances in the good bacteria in there which allow the bad guys to move in. So we've got pathogenic bacteria, amino acid derivatives. If we don't break our amino acids down, we get these volatile chemicals produced. And we need to look at prebiotics, which is the food for probiotics. So basically, in a nutshell, you're looking at the enzymes that digest our food, the pathogens, to remove the pathogens, replace the enzymes, and look at probiotics and prebiotics. And that way we'll see we build up a sort of pattern of how to get in and diagnose gut problems. So we often use those 2 points for the gut. You usually find there's a therapy localization of one or the other. Just to refresh there, you see the transverse, that in many people this sags and can sag even more than that, And you can see you get more than a right angle bend then at the hepatic and the splenic flexures. So you see there that why you get a build-up of wind and things. If your colon here is fermenting, in other words, you're not digesting all your food, and you've got undigested proteins down there, you've got fungus eating all the sugars and everything down there, you'll get a build-up of wind in there before the stricture, and that'll blow this, so it'll get bigger, and that gives you pain. So the two ways you get abdominal pain is colic, which means that the circular muscles become constricted or in cramp, and distension. Those are the two ways you get abdominal pain. You don't usually get pain from anything else unless you've got an acute appendicitis or infection. So the two ways you get pain in there is colic, in other words, spasm of the muscles, and obviously if you get spasm of the muscles, you then get a build-up of feces and air, bubbles and gas. So the gas comes more proximal to the constriction, but you may not have a constriction. You could just have a lax transverse colon with a bigger bend in there, and of course gases and feces are hard to get around those bends, just like water pipes or waste pipes, really. So let's have a look at the stomach. The stomach produces a pre-enzyme called pepsinogen, which will convert with hydrochloric acid to pepsin, the activated form, and also produces a thing called intrinsic factor. Now, it does produce a number of other small hormonal-like substances which act within the stomach itself, secretin, etc. But we want to talk about the main ones, which is hydrochloric acid and pepsin. So those would be the things that you would need to use as biomarkers: hydrochloric acid, pepsinogen, the precursor, pepsin itself. So hydrochloric acid activates pepsinogen into pepsin. Pepsin, its main function is to start the breaking up of proteins, which are long-chain amino acids, into smaller ones called peptides or polypeptides, so they become smaller chains. So the first job of pepsin is to digest proteins. So the last thing you want to do is to give, as a supplement, pepsin if a person doesn't need pepsin, because they'll start digesting their stomach, okay, and they get pain. In other words, the stomach will start eating itself. Do not ever give pepsin if you don't need pepsin. If a person needs pepsin, then fine, it's good to substitute it, supplement it in for a while. But don't give pepsin. And this is why most companies will give hydrochloric acid or betaine hydrochloride as a separate product, and betaine and pepsin for those who need it. So it's very important that you muscle test which one a person needs, because you don't want to give pepsin. Now, if you've got a deficiency of pancreatic enzymes, say, and all enzymes are proteins, okay? So if you need to get pancreatic enzymes to a person, you need to give the pancreatic enzymes. You don't give it with pepsin. So to give a person a digestive enzyme mix, which includes pepsin, if they don't need the pepsin, that pepsin will digest the other enzymes, so they won't actually really work. They'll digest the protease and the amylase and the lipase enzymes, which are needed further down. So if you need lipase, give lipase. If you need amylase, as we'll see a lot of people do, give amylase. Don't give it with pepsin, because it'll never get there. It'll digest in the stomach. So this is important that you work that out. So this is a stomach lining. Here's the stomach wall, here's the lumen there. And this is a chief cell, they're called here, which secretes the pepsinogen. So pepsinogen is not active, because we don't want to make active enzymes. This is why nature has given us the ability to activate or not. So you need to have the substrate there, which is the pepsinogen. So this is the pre-enzyme, if you like, and it needs the addition of hydrochloric acid to convert it to pepsin. So it's a feedback mechanism. So when you take protein into the gut, you then secrete hydrochloric acid, which activates the pepsinogen into pepsin. So the hydrochloric acid comes from the parietal cells. So this is the cell which produces hydrochloric acid. But this parietal cell also produces a substance called intrinsic factor, which its job is, again, it's a peptide, and it locks onto vitamin B12. So cobalamin within our diet of B12 from animal foods usually has this intrinsic factor locked onto it, which enables it to be absorbed further down in the small intestine. So if you have a failure of intrinsic factor, you will then get poor B12 absorption, and then you'll eventually end up with B12 deficiency with anemia, megaloblastic anemia, or pernicious anemia it used to be called. So low B12 could be simply that you're not secreting intrinsic factor. So if there's a problem here, you may produce low hydrochloric acid, but the more important thing here is that low hydrochloric acid probably means that you've got low intrinsic factor. So you need to always test a person that they could end up with B12 deficiency because they got a stomach problem. Okay? So the question is, if a person has low hydrochloric acid, they then won't activate pepsinogen to pepsin, so they would be deficient in pepsin, then they would have poor breakdown of their proteins, and they will get all sorts of symptoms of what is going on in the stomach. In other words, they get a lot of wind because they're not digesting the protein, it starts fermenting in there, and then they get burping, belching, and all the other symptoms that are characteristic of low protein digestion. And they may get also allergies, because they'll start further down, maybe absorbing longer chain proteins, which are thought by the body to be foreigners, instead of digesting them right down into single amino acids. So people can get multiple allergies. Dr. Goodart always used to give hydrochloric acid, particularly, or hydrochloric acid and pepsin, to people with allergic problems from food that they eat. So the other factor is that if the parietal cell produces intrinsic factor and hydrochloric acid, you need to therefore, if you're going to supplement B12 in as cobalamin, hydroxycobalamin, or in any other form, adenosylcobalamin, methylcobalamin, you need to do it when there's hydrochloric acid in the stomach, because that's when you produce the intrinsic factor. So they come out of the same cell. So this is why the time when you produce the highest hydrochloric acid is at breakfast time. That's why it's called stomach time. So at breakfast between 7 and 9, during our standard time zone, is when we produce more hydrochloric acid. So it's a very good time to have protein. So people who skip breakfast, this is the time when you probably will absorb your protein better than any time else. So if you have a high starch meal and low protein at breakfast, then you've got a bigger chance you're going to be hungry later on, because you haven't got the protein. So if you have the protein, you tend not to get hungry later on. So you need some form of protein at breakfast. So remember, therefore, the optimal time, which the muscle testing will tell you anyway with the body clock, to administer B12 is always at breakfast time, because that's when we produce the intrinsic factor. So we know that hydrochloric acid acidifies the stomach. Basically, the stomach is producing acid all the time. In fact, it produces acid highest before we have a meal, because that's what tells you you're hungry. It's when you produce the hydrochloric acid and starts to activate the pepsinogen that you feel your stomach's going to digest itself, so you know you've got to eat, okay? And that eating is what then satisfies the hydrochloric acid, and the pH of the stomach actually goes down. So before a meal, it could be 1. So if you swallow anything before a meal, you're going to have a very high acidity in the stomach. This is the fallacy of taking probiotics. Okay, if you take probiotics with or just before a meal, you will destroy them very, very quickly, because one of the functions of hydrochloric acid is to destroy bacteria and parasites. That's why, another reason why we have it in there. So the only way to get a probiotic through the stomach is to take it on a time when the pH is less acid, which is usually when you go to bed, at triple warmer time, 9 to 11, the stomach acid is less, or preferably in a delayed release capsule, which bypasses the pH and only breaks in a pH of 6. Anything more acid than that, is preserved. So you need to, with probiotics, as we'll see, really deliver them into where they're needed, which is the small intestine and the large intestine. Anything that goes through the stomach potentially as a bacteria will be destroyed. So it acidifies the stomach, which the main reason for that is to activate pepsin or pepsinogen. It destroys the bacteria and parasites left in food. And it also ionizes, because it makes the contents of the stomach more acid in water, which splits or dissociates minerals. So minerals, when we take them in our food, are in compounded or locked form, solid form. So let's say sodium chloride, which is salt, is sodium chloride bonded together. If you put sodium chloride into water, it becomes dissociated. In other words, the sodium and the chloride split. In other words, it becomes ionized. The sodium has a positive, the chlorine has a negative. And this is what happens when minerals go into water. And obviously something which is 100% soluble, you won't see any solid mass in there. So this occurs because we produce stomach acid. So minerals will dissociate in an acid medium. So that's a major function of hydrochloric acid in the stomach, is to make sure that we produce enough hydrochloric acid, otherwise you can end up with zinc, magnesium, calcium, etc. deficiencies. Now we need to look at some stomach-related muscles, and these would be the ones that we would choose. So we have pectoralis major clavicular, and particularly if it's bilaterally weak, traditionally in applied kinesiology, this is a sign of low hydrochloric acid in the stomach. If it's unilaterally weak, it's certainly the stomach involved, or could be. Sternocleidomastoid, along with the neck flexors. Biceps brachii, brachioradialis, pronator teres, pronator quadratus, the opponens pollicis and the opponens digiti minimi. These are all stomach-related muscles that we can use and test a person potentially for a stomach problem.